Hi guys. It is Wednesday. No, it is Monday. Monday, October 12th. How can you hear me? Can you hear me well?
Yes.
Thank you. Thank you. So our third and last question on, last lecture on enzymes. On enzymes, so you're supposed to be seeing my slides. Can you see my slide?
Yes.
Oh, yeah, okay. So let's finish our discussion. Actually, we're gonna finish it last time, but just wanna remind you about this ratio of kcat over km, right? So remember, kcat is a turnover number, basically the catalytic rate constant. The higher it is, the faster the reaction, regardless of what the rate determinant step is, right? And km, remember, Michaelis constant. And remember, Michaelis constant, basically, the lower it is, the higher the affinity of the substrate for the enzyme. So you want kcat to be high, and you want km to be low. In other words, you want this ratio to be high. And remember, I told you that this was the measure of kinetic efficiency of an enzyme. And there is unfortunately a limit, right, as far as how efficient enzymes can be. And that's due to diffusion, right? I told you no matter how efficient the enzyme is, if the substrate is not floating by, if the enzyme is swimming in a soup of big molecules, small molecules, but never encounters the substrate, then the enzyme cannot perform the reaction, right? So the substrate does not diffuse to it. And so this number is limited, 10 to the 8, 10 to the 9, and the enzymes that have achieved that, we say that these enzymes have achieved catalytic perfection. And you can see one of the most efficient enzymes is acetylcholine esterase. So if you remember this enzyme, what it does, what's acetylcholine? Anybody remembers?
Neurotransmitter.
Neurotransmitter, correct. So, right, so basically it's a neurotransmitter which passes the neural signal between the two neurons, or is present at the neuromuscular junction, right? So it's released into the synaptic cleft by the presynaptic neuron. And then there is, once the signal has been transmitted, acetylcholine esterase comes in and chops it up, removes the acetate, releases the choline molecule, and the choline goes back into the presynaptic neuron, ready to be acetylated and ready for the next what do you call that? Nerve impulse, right? There's a word for it. Yeah. Action potential. So you can see for acetylcholine, both kcat is high and km is low, and that results in a very high kcat over km number. But some enzymes, for example, such as catalase, you can see here the km is not very low, right? So in other words, the km for catalase, it doesn't bind hydrogen peroxide strongly, but it compensates for that with a very high number of kcat, right? So if you divide this by that, you still get a pretty high number overall. So it's a ratio of kcat over km. It's not just one parameter. It's the combination of the two that will tell you about the catalytic efficiency of the enzyme. Okay, any questions? All right. So let's talk about enzyme inhibition. Yeah, so after all, many drugs have been developed that work by inhibiting enzymes. Since the enzymes are so important as biological catalysts, right? So many enzymes are overexpressed in diseased tissues. In cancer tissue, many enzymes are overactive. So if you want to inhibit an enzyme, you need to know how to do that, right? And so you need to basically be familiar with different types of inhibitors so you can develop different kinds of drugs. Well, first of all, let's think about the reversible inhibitors, right? So these are molecules which will permanently form a covalent bond to one of the amino acids at the active site, right? And basically, these are very powerful molecules. You just need one. You just need one molecule. With one molecule, small molecule, you can kill an enzyme. The problem with such molecules is that they're extremely reactive, right? And they're toxic. So if they inactivate the enzyme, they can also inactivate other biological macromolecules in the body, and that can lead to toxicity. So when you develop an irreversible inhibitor, you really have to be careful. You really have to be careful that to make sure that your inhibitor specifically reacts with the enzyme in question. Right, and pharmaceutical industry has been careful, even though there are a lot of irreversible inhibitors on the market. Can you name one of them? Can you guys name the most famous irreversible inhibitor? Something that inactivates an enzyme, kills an enzyme?
Is it like proton pump inhibitors, or what is it for the stomach acid?
There is a proton pump inhibitor, which is irreversible. That's correct. Can't remember the name, but yes, they're sulfoxides, and they react irreversibly with the proton pump that pumps out protons from the gastrointestinal tract.
Would radioactive iodine be in that same group? They use it to basically kill the thyroid gland, or is that not the same?
Well, you just damage, right? So you use the radioactivity to damage. So it's a different mechanism, right? So it's not that you are covalently inactivate any kind of enzyme. You just basically damage the tissue with radioactivity, right?
I believe so.
Yeah. How about when you have a headache? What do you take? Or high fever?
Tylenol?
Well, how about an older drug before Tylenol?
Would aspirin be it?
Aspirin, that's right, aspirin. So aspirin is one of the oldest drugs, and it is one of the safest. That's most surprising, right? So even though everybody is paranoid about these irreversible inhibitors, thinking that they're so toxic, but aspirin has been around for over 100 years, and it's one of the safest drugs there is, right? So all right. Now, reversible inhibitors, on the other hand, they bind at the active site and they can dissociate from the enzyme, right? They come and go. But by coming and going, they block the substrate, they block the site where a substrate normally would bind. And so the substrate will have limited possibility to bind at the active site. And in most cases, these reversible inhibitors, they are structural analogs of substrate or products, right? So basically they look like substrates. And so the enzyme thinks that this is a substrate and will accept it as a substrate. But then the reversible inhibitor will just sit there, occupy the site, and no reaction will occur. And so these are often used as drugs to slow down a specific enzyme. So obviously these reversible inhibitors can bind to free enzyme and prevent the binding of the substrate, which is what I just told you. But they can also bind to the enzyme substrate complex and prevent the reaction. So this happens if the reversible inhibitor binds at some other site on the enzyme, right? So the enzyme has already bound the substrate and just about to undergo the reaction, but here comes the inhibitor, binds somewhere else on the enzyme and prevents some kind of conformational change that the enzyme is about to undergo to perform a reaction. And that will slow down the reaction or prevent it. All right, so these are some main mechanisms, how inhibitors work. And so let's look at 3 different kinds of inhibition. So there is a competitive inhibition. So where basically inhibitor competes with the substrate for binding. So it binds to the active site and it does not affect catalysis. So here's our enzyme, here's our substrate, and here's our inhibitor. So inhibitor will bind the enzyme and shift the equilibrium towards enzyme inhibitor complex. And so because of that, the enzyme will be tied up with the inhibitor. And that, based on Le Chatelier principle, will move this equilibrium to the left. And thus there will be only small amount of ES left for the reaction to occur. Okay, so here's the graphical representation of that. So the enzyme will bind the substrate at the active site, but the inhibitor can bind to the enzyme at the active site as well and block the active site, right? So the substrate can no longer bind to it. Well, there are a couple consequences to that. One is that there's no change in Vmax. Just think about it. So overall, you know, if the substrate can actually outcompete the inhibitor. Let's say you freeze the inhibitor, let's say at 2 micromolar, but you just keep increasing the amount of substrate, more substrate, more substrate, more substrate. Eventually, you'll start moving this to the right with more substrate. More ES will form and more reaction will form, and eventually you can achieve Vmax, right? So by just adding more substrate, you can you don't change the Vmax, but you can reach it, right? You can reach the Vmax with your reaction rate. And secondly, there's apparent increase in Km. So remember, Km is the measure of affinity of the substrate toward the enzyme. And so by adding the inhibitor, you have this apparent increase in Km. So in other words, it's not that this enzyme has lost the affinity for the substrate. It's just that there's less of enzyme available to bind to the substrate because it's tied up in a complex with the inhibitor. And that's why you have this apparent increase in Km, even though Km has not changed. But the term for Km may have changed. So in other words, you think it has changed, but it hasn't. And that's actually, we can see that. Let's redraw the Michaelis-Menten equation. So this will be Vmax substrate concentration. alpha Km plus substrate concentration. You can see it's this term where Km usually is going to be increased due to the alpha coefficient. And you can see that alpha keeps increasing the more inhibitor you add, right? I is the inhibitor. The more inhibitor you add, the larger is this term. And if we go to Lineweaver-Burke plot, you can see here, take the reciprocal, the slope will increase. The slope will increase because of this parameter, alpha, right? Alpha 1, alpha 2, alpha 3. Now the y-intercept, which is this point here, it will not increase or change in any way because it will still be equal reciprocal Vmax. Reciprocal Vmax. So, so this will be the case of competitive inhibition. Are there any questions about this? All right. So remember, for competitive inhibition, the change is primarily in the Km term. The Vmax does not change. So the moment you hear that the inhibitor does not affect the Vmax, that should tell you, most likely I am dealing with a competitive inhibitor. Okay, the second out of 3. So uncompetitive inhibitor. So this will bind to the enzyme substrate complex and does not affect substrate binding. So instead it inhibits catalysis. So the enzyme will come in, bind the substrate, it will form the enzyme substrate complex, right? Which can go to the product. Alternatively, it can be trapped. It can be trapped by the inhibitor, like so. Trapped by the inhibitor to form this ternary complex, which has all 3, enzyme, substrate, and inhibitor, and from which there will be no reaction. No reaction. So, so the way this will normally work is that the substrate will bind first, occupy the active site, the inhibitor, and before the reaction can occur, the inhibitor will come in and bind at some other site on the enzyme to form the ternary complex with the substrate, the inhibitor, and no reaction will occur. So this will decrease Vmax, right? So basically it slows down the maximum reaction. And there will be no change in the slope, and we will see that in a moment. And Lineweaver-Burke lines are parallel. So let's look at those. So the Michaelis-Menten equation takes on this form. Takes on this form, plus this time it's going to be alpha prime times substrate concentration. Now, if you transform this to Lineweaver-Burke plot, so you can see here the slope does not change. The slope does not change. What will change is the y-intercept. y-intercept. You can see here, as you increase the inhibitor concentration, right, the y-intercept will increase. So this is alpha prime 1, alpha prime 1.1, alpha prime 2. So y-intercept will increase, but the slope will not increase. And so this is y-intercept, alpha prime over Vmax, right? And if you can do some algebra to it, you will find the x-intercept, right? And so this is the case of uncompetitive inhibition. So here, when you do the, if you have a problem, right, usually you'll be given substrate concentration, V naught. And if you find that different inhibitor concentrations, the slope does not change, then it's a sign that you're dealing with uncompetitive inhibition. But you may have a question. Now combine this. So let's say, oops, I already opened my hand. So competitive inhibition, where the slope changes, y-intercept doesn't. Uncompetitive, when the slope doesn't change, but y-intercept does. So now ask me your question. Ask the
How
question.
does the slope not change if Vmax changes?
Vmax changes, but Km changes as well. So the ratio of Km over Vmax stays the same. Okay? So you can see here, I guess, as you change the Vmax by this number, all right, you change Km by pretty much the same number, right? So the ratio stays the same. Did that answer your question?
Yes, I did. Thank you.
Mm-hmm. All right. So, all right. So the question is that I wanted you to ask me. So there is also a possibility for mixed inhibition. Mixed inhibition is when actually both types of inhibition, competitive and uncompetitive, are combined. Sometimes this mixed inhibition is also called non-competitive. You may see this in the literature. Non-competitive. So both possibilities are present. So we have enzyme and substrate. The enzyme can bind the inhibitor to form inhibitor-enzyme complex, and the substrate is all left by itself, no active site to bind to. And alternatively, the substrate can bind to the enzyme and can undergo reaction, but can also be intercepted by the inhibitor, which will bind at the different site on the enzyme. Okay, so here's a possibility. So here's enzyme binding the substrate and the enzyme substrate complex binding the inhibitor. There's also a possibility for the enzyme binding inhibitor, which will then bind the substrate. And so what's important here is that both of them will bind. So the inhibitor will bind with or without the substrate, right? And it will bind to a regulatory site. So remember regulatory site. So these will be allosteric enzymes, right? And so the inhibitor will affect the substrate binding, and it will also affect the enzyme rate. So you can see it will decrease the Vmax, and it will also change the Km. Now, how it will change the Km is not clear, right? Depends on the enzyme. Can increase it, can decrease it, depending specifically how it changes the conformation, right? All right. So here's what the plot will look like as you increase the inhibitor concentration. Now, let me redraw the Michaelis-Menten equation. Vmax. So here we have both coefficients, alpha and alpha prime. One of them is present in the slope. The other is present in the y-intercept, right? And you can see what's happening here. So the slope changes, and the intercept is somewhere over there off the y-axis, right? And so the y-intercept will change depending on the alpha prime. And depending on the alpha, the slope will change. So, and this will be no inhibitor. So this is the third type of inhibition, right? So all three are present in enzymes. So, obviously when you discover drugs, right? So the kinetic expressions are crucial because if it tells you it tells you where the inhibitor binds, right? So let's say if you do Lineweaver-Burke plot, and then you get something like this, right? If you increase the inhibitor concentration and then you get the slope changes, but the y-intercept doesn't change, you can say, okay, my drug molecule that I just discovered binds at the active site. If I get something like this, you can say my drug molecule binds somewhere else on the enzyme. I don't know yet where, but that's the subject of my further research, right? Any questions? Now, there's a third type I mentioned to you, irreversible, right? So let's say if I have enough time to show you irreversible inhibition, I probably do. If I don't, I'll just give you some pages to read from the textbook. Okay, so so oftentimes these irreversible inhibitors. Now I'll send you. You don't have these slides. I'll send you these slides. So just listen for now. I'll send you these slides. I mean, I will put them in the files on Canvas. So oftentimes these irreversible inhibitors are known as enzyme inactivators. And their structure, so structure similar to substrate or product, right? So they can actually occupy the binding site or the active site. And generally they form a covalent bond. That's why they call it reversible, right? Because once they form a covalent bond, they don't come off. But it's not required. So in other words, some inhibitors form such strong non-covalent bonds that they almost like covalent. So they will never come off the active site. And so because of that, it's not necessary to maintain the inactivated concentration once the enzyme has reacted. So complex cannot dissociate. So in general, when you have a drug that is reversible inhibitor and a drug which is irreversible, now can you predict the dosage regimen? Like let's say if you were to prescribe, you were a doctor and you didn't know about the, well, you should know if you're a doctor, but let's say you, in a difficult situation, you don't have access to your computer and you need to predict the dose for a particular medication to give it to a patient. If you have a reversible inhibitor, will you increase the dose or decrease the dose compared with irreversible? So reversible over irreversible. 3 times a day or 1 time in 3 days?
Would it be 3 times a day
For which one?
for reversible?
Reversible, that's right. So reversible comes on and goes off, right? You always need to maintain the concentration of reversible inhibitor high so it can compete with the substrate for the active site. For irreversible inhibitor, you don't, right? So you basically inactivate the enzyme and then the enzyme. Now the question for you, is the enzyme totally dead? So once you inactivate the enzyme, what happens? So that does it mean that you die too because you don't have this enzyme in the body anymore? What do you think?
Can you make new ones?
Yes, you will make new ones. That's right. So the enzymes which have already been made through genetic machinery of the cell, those are dead, but the genes are still present, right? The genes are still present. There will be more gene expression, more proteins will be produced, and more enzymes will be produced. That's right. Okay. Right, so reversible inhibition does not mean permanent. Gene encodes for other, so the gene will encode for other enzyme copies. That may take hours, sometimes days. And so because of that, actually, irreversible inhibitors are not so bad as some people think. In fact, 1/3 of enzyme targets have a reversible inhibitor drug available. And here we were told about the proton pump inhibitors. So for example, esomeprazole is one of the best-selling drugs on the market. I think it's so it's one of the top-selling drugs, and it is actually an irreversible inhibitor. So 2 different kinds: affinity labeling agents and mechanism-based enzyme inactivators. We'll look at both. So here's an affinity labeling agent. So you have enzyme and inhibitor, some kind of binding constant. So first it forms a reversible complex, and then the reaction can occur and you form a covalent bond. And generally this will be some kind of alkylation or acylation. And so in most cases, the K inactivation is the rate determinant step. It's this one, right? And so the time-dependent loss of activity occurs. Okay, so the affinity labeling agents are generally reactive, okay, and therefore potentially toxic. So many of them react with other proteins and DNA. So basically, these are general alkylators, right? So for example, how about this guy? Ethyl bromide. Highly toxic, right? So if you have a nucleophile, any kind of nucleophile, this will undergo an SN2 reaction. And your nucleophile, what if your nucleophile is DNA? You get DNA damage. You get DNA damage, you get cancer, you get any sorts of genetic disease, and so on and so forth. So this will be an example of an affinity labeling agent. And so it still can be effective because once the complex forms, the reaction in essence is unimolecular, right? So basically more rapid than non-specific bimolecular reactions. So in other words, once it binds to a specific target, then the reaction becomes unimolecular. Remember, unimolecular reactions are faster. And also in your enzyme that you're targeting, there must be some kind of nucleophile present at the active site, right, near the reactive group. So, which means that your enzyme that you're targeting has higher chance of reacting with your affinity labeling agent than other enzymes. And in the case of anti-tumor agents, DNA precursors rapidly transported and concentrated at tumor target. Right, so basically, for example, your affinity labeling agent can be masked as a DNA precursor. And so because DNA divides, right? So a lot of DNA precursors are rapidly transported into the nucleus. And so if your affinity-based reagent is part of that DNA precursor, then you can make it somewhat selective. All right, and so for design of such an effective affinity labeling agent, so obviously you want to decrease the Ki, which is the dissociation constant of the inhibitor from the enzyme. You want to modulate reactivity and incorporate reactive group so it is near the nucleophile in the active site of the target enzyme. And usually that comes from X-ray crystal structure, right? So for example, here is your enzyme. Here is your enzyme. Here is your nucleophile, some kind of serine. Here is the active site, and you want your affinity labeling agent to occupy the active site in this way, so that the serine can attack this carbon and this can leave, right? So this comes from the X-ray of the enzyme from the enzyme substrate complex. So if you know that these are in close proximity, then you can say that, oh yeah, my affinity labeling agent is going to be a selective molecule. Any questions, guys? So these are irreversible inhibitors. Let's see, I have maybe a couple more slides here. I'll skip this one. So some of the probably, so I told you about aspirin. I'll have one slide on aspirin as well. But for example, penicillins, right? Penicillin is a one of the first antibiotics ever discovered, right? So here's the structure, and you can see there are different kinds of penicillins. So when usually people say penicillin, that is incorrect because there are actually many different penicillins, right? And penicillins change structurally depending on this group R, right? For example, if the group R is benzyl, it's penicillin G. If the group R is this phenoxy methyl, it's penicillin V, right? Then we have ampicillin, amoxicillin, and so on and so forth. Anyway, but the whole point of these penicillins is this beta-lactam ring. Okay, it's a 4-member chain with the nitrogen, oxygen, like so. And this ring will undergo ring opening. So remember, 4-member rings are strained, right? 4-member rings are strained, and they will undergo ring opening. Now, there is a related type of antibiotics known as cephalosporins and cefamycins, but they also beta-lactams. Structurally somewhat different. All right, so we called penicillins and cephalosporins as ideal drugs. And specifically, the reason for that is because they inactivate an enzyme essential for growth of bacteria, but does not exist in animals. They're bactericidal, they kill bacteria. Remember, all antibiotics can be divided into 2 groups. There are bactericidal and there's bacteriostatic. Which one do you think is better, bactericidal or bacteriostatic?
Bactericidal.
Why don't you like bacteriostatic?
Bacteriostatic just stops the growth. Bactericidal actually kills bacteria.
So if you stop the growth, that's not bad. No?
No, but I don't think it actually kills whatever is currently present.
Well, if you stop the growth, it doesn't proliferate and doesn't kill you, right?
So wouldn't you want to use a bacteriostatic when it's like your body or something? If it's like a surface, you would use a bactericidal?
Yeah, well, I mean, bactericidal sounds better indeed because you kill the bacteria, but many antibiotics are actually bacteriostatic and they do work as well. And
that's what I'm saying. Like if it's bactericidal, it's most likely not going to be used on your body because it might be toxic or something.
Well, beta-lactams, so the penicillins, they're bactericidal, and they're not toxic, right? So you can actually take a very large dose of penicillin. You may have some allergic reaction to it, but it will not kill you because of its mode of action. What bacteriostatic allows you to do, it slows down the growth of bacteria to the point that the organism can actually handle the bacteria on its own. Right, basically, it kind of gives it gains extra time for the organism to mobilize its immune defenses, and then you actually destroy the bacteria. But bactericidal, yes, basically it kills the bacteria right away. You don't need to wait for the immune system mobilization. All right, so it inactivates. Go ahead.
So is that better because you want your to trigger immune response to use bacteriostatic? Is that why you rather use that?
Well, with bactericidal, you also trigger the immune response. You know, you kill the bacteria, but it'll still be in your system for a while, right? All the particles, but bacterial particles will still be in your bloodstream for a while.
Mm-hmm.
So you'll still have antibodies against them. Yeah, I don't know. Yeah, so there are advantages and disadvantages to both, and both are used in the clinic. So So the penicillins inactivate this enzyme known as peptidoglycan transpeptidase, and this blocks cell wall biosynthesis. Now, remember bacterial cell wall, so which is present in gram-positive organisms. Shall we? Is cell wall present in gram-negative too?
Yes.
Yes, correct. Yes. Cell wall. Gram negative. Well, not easy to Google. It says composed of a thin peptidoglycan cell wall. So that's why I was a little confused. I guess it does have cell wall, but it's a thin one. Okay. Anyway, so so peptidoglycan. So you can see the reason why there's peptidoglycan cell wall in bacteria is for bacteria to withstand osmotic pressure, right? So bacteria are supposed to survive in a variety of different extremes. And so they all, because these are unicellular organisms, they often end up in a situation when there's a severe osmotic pressure. And so what happens is that they need to put together this cell wall consisting of glycan, which is the chain composed of sugar, like N-acetylglucosamine and N-acetylmuramic acid, and a little peptide here shown here, which will undergo cross-linking. So there's this enzyme, peptidoglycan transpeptidase. And so there's a serine residue here on this enzyme. And the serine will attack the carbonyl, and this will be the leaving group. So this is D-ala. So it's a D-alanine. So very unusual. Remember, most amino acids are L, but in bacterial cell wall, this will be D-ala, D-alanine. So this will leave and to form the transient ester here, and the lysine will come in and relieve the enzyme. So basically, they'll form the amide. So now you have the amide from the ester with the enzyme. And so without the cell wall, high internal pressure causes bacteria to burst. So here's an example of without the cell wall. So this is normal bacterial cell division. And so this is bacteria treated with the fosfomycin, which also destroys the cell wall. And you can see here, it just bursts and all the contents spill out, right? And so it turns out that you can see here when the peptidoglycan is being cross-linked, you can see here you have one D-ala. This is D-ala. And then you have a second D-ala. Second D-ala. And if you look at how this, at the structure of this, and look at penicillin, you can see the similarity, right? Nitrogen, carbon, carbonyl, nitrogen, carboxylic acid, methyl, and so on and so forth. So the enzyme thinks that penicillin is D-ala, right? And it binds penicillin thinking it's D-ala. And what happens is that once it's bound penicillin, this serine here I was telling you about now attacks the carbonyl and opens the 4-member chain to form this ester, right? And so this is a highly facile reaction because 4-member chains are highly unstable, Remember the instability, right? 3-membered least stable, 4-membered still quite unstable. And so beta-lactams undergo ring opening, and you form the covalent intermediate, covalent complex, basically between penicillin and the serine residue on the enzyme. And so the reaction is blocked. Reaction is blocked, and the bacteria can no longer complete the synthesis of its cell wall. Does it make sense? So you can see penicillin is another example of an irreversible inhibitor. And I was telling you about aspirin. So aspirin now originally was even mentioned. The willow bark for pain was already recommended by Hippocrates in the 4th century BC, right? And sodium salicylate was isolated from willow bark, was recommended for rheumatic fever, 1875, and Bayer then actually made the actual aspirin as we know it. Sodium salicylate is the compound without the acetate, but it had very strong gastrointestinal effects. And so by adding acetate, those side effects were reduced. All right. Any questions? So these are irreversible inhibitors. I'll put this up to Canvas. Okay, so let's look at so to finish this chapter, there are a couple other topics. Specifically, I want to talk to you about some of the mechanisms, how the enzymes work, some specific organic chemistry mechanisms, right? So with electron pushing. So we'll look at the digestive enzyme chymotrypsin, right? So basically it's an enzyme that belongs to the family of proteases. So it's a protease, so the enzyme that breaks down proteins, and it cuts the peptides at specific locations on the peptide backbone. So chymotrypsin chooses the locations which are close to large aromatic groups. So tyrosine, phenylalanine, and tryptophan are the usual sites where the peptide bond will be cleaved. So chymotrypsin will cleave it right here. Okay. And so the way it's going to happen, so there is a what's known as a catalytic triad. So aspartate 102, histidine 57, and serine 195. So this is referred to as a catalytic triad. Catalytic triad. And another important thing here is, so this is red. Let me choose a blue color. So this NH and this NH, from glycine and serine, they form what's known as oxyanion hole. Oxyanion hole. Basically, it's a type of a hole which stabilizes negative charges through hydrogen bonding. And there's also hydrophobic pocket here, right? Which accommodates large aromatic residues. And here's the substrate. Here's the substrate, which will bind at this active site. Okay, so how does this cleavage work? So here is chymotrypsin. It's a free enzyme. Now here's the catalytic triad. So we have aspartate 1 or 2. It forms a hydrogen bond with the imidazole ring of the histidine. Now, as it does so, note the electrons. The electrons will move over towards this nitrogen, right? And towards this hydrogen. Towards this hydrogen, and that will create a partial negative charge on this serine. Partial negative charge on this serine, making serine into good nucleophile. Good nucleophile. Now, the substrate comes in. Let me turn this into red. Substrate comes in, occupies the binding site, and now you can see that when the imidazole from the nitrogen grabs the proton, serine electrons are released. It will attack the carbonyl on the substrate, okay? And the electrons will move over to occupy this area. This area, remember, is the oxyanion hole. Oxyanion hole. So, and once that has happened, then we have basically a tetrahedral intermediate, right? So the new bond has formed. So this serine oxygen and the carbon of the carbonyl bond has formed, and the carbon-nitrogen bond of the peptide is still there, right? So we have a tetrahedral intermediate in which the O- is stabilized by the 2 hydrogen bonds from serine and glycine, right? So this is an oxyanion hole. And keep in mind that this hydrophobic residue is holding this whole thing together, right? So basically it's stuck there, it's like an anchor, freezing the peptide in this particular orientation at the active site. Okay, so if you have any questions, just keep asking me, okay? All right, so then in the next step, the tetrahedral intermediate will collapse. The electrons from the oxygen will come back down, right? But now this time, instead of the carbon-oxygen that bond that breaks, it's carbon-nitrogen bond that breaks. And this grabs the proton. And so we have the first half of the right-hand side of the peptide leaving, right? So this red hydrogen is that red hydrogen, right? And we have this transient enzyme substrate intermediate. Let's write this down. Transient enzyme substrate intermediate. So now we have this ester, but so half of the peptide is still attached to the enzyme, right? So this is transient intermediate. Now we have to break this ester to release the left-hand side of the peptide. And so this is what will happen in the next step, right? So water will come in, it will hydrogen bond to the imidazole, and then this oxygen will gain partial negative charge, right? Partial negative charge. And this oxygen will attack the carbonyl of the ester. And again, the electrons will move over into the oxyanion hole. So this oxyanion hole, just like, accepts electrons, releases them, accepts, releases. So the enzyme has evolved for, to gain this ability to keep stabilizing this negative charge that keeps coming, keeps going, keeps coming, keeps going. And so once this tetrahedral intermediate is formed, now this oxygen has a full negative charge. It's hydrogen bonded to the two nitrogens, right? And when the electrons come back, this bond breaks and it retains the hydrogen, which will be attached to the serine, and the imidazole would leave, like so. So the serine will basically regain its hydrogen, and now it's hydrogen bonded to imidazole. And so the left-hand side of the peptide is now still sitting here in the oxyanion hole, but it will dissociate. It will dissociate on the next picture. It will dissociate and release the free enzyme. So chymotrypsin is ready for the next molecule of the protein. Any questions, guys? So basically this enzyme works through a bunch of the tetrahedral intermediates, right? There's a catalytic triad is another important thing to pay attention to. There's oxyanion hole, catalytic triad, oxyanion hole. There's hydrophobic pocket for aromatic amino acid, right? And obviously catalytic triad makes the serine nucleophile much more nucleophilic. And the last type of enzyme I want to show you is lysozyme. So lysozyme is present in tears, present in saliva. Basically, it's an antibacterial enzyme. And it destroys the bacterial, also peptidoglycan, okay? Also destroys bacterial cell wall. So the glycan is shown here, okay? So you can see here, basically a series of sugars connected through ether linkages, right? And so the sugars are alternating residues of what's known as N-acetylglucosamine, NAG, and N-acetyl muramic acid, NAM. NAG, NAM, NAG, NAM, NAG, NAM, NAG, NAM. And the cleavage will occur at this site here, okay? And this is the inset. you can see what's going to happen is the lysozyme is going to break this ester linkage into two alcohols. So it's a hydrolysis reaction, which is catalyzed by the enzyme. So there is an X-ray of lysozyme with the bound substrate, whole bunch of them actually, different analogs, which show that actually the C1 carbon, now C1 carbon, it's right here. This is the C1 carbon. C1 carbon is, no, wait. Yes, no. Does it have a, hold on a second, guys. Oh, this is C1 carbon, I'm sorry. This is C1 carbon. Like so, right? It's attached to the oxygen in the ring and attached to the oxygen outside the ring. And so it has been shown with the X-ray structure is that it's has glutamate and it has aspartate as the key amino acids at the active site. And so we're going to skip the slide because basically a picture is worth 1,000 words. So there are two mechanisms that have been proposed. So one of them is what's known as an SN1 mechanism. The other is SN2. So in the SN1 mechanism, so glutamate 35 will protonate this oxygen. This oxygen will kick out the ether oxygen to generate the carbenium ion, right? So this is a carbenium intermediate where they have a positive charge on this carbon. Let's go to the next slide. We're going to look at SN2 next. So once that happens, water will come in. Water will form a hydrogen bond with glutamate 35, and then the oxygen on the water, which will be partially negatively charged, then can attack the carbon of the carbenium ion to form the alcohol. And so and regenerate the two amino acids, glutamate and aspartate. The second mechanism, in which actually aspartate 52 now plays a role as a transient, again, nucleophile, right? So again, the aspartate will attack this carbon. This will be the leaving group. The leaving group will grab a proton from glutamate 35 and form a new bond, shown here. Form a new bond, so it's going to be a covalent intermediate. And then glutamate 35 will grab a molecule of water, which will come in from the top and release the aspartate 52 to regenerate alcohol and the glutamate and aspartate. So the difference is, again, between SN1 and SN2. So there has been a lot of debate in the literature as far as which mechanism is more credible, right? And lots of different ways to elucidate mechanisms have been employed. One of them, for example. And so what's the main difference in stereochemistry between SN1 and SN2 mechanisms? Who remembers from OChem 1? Stereochemistry of SN1 and SN2.
I think there's no change in stereochemistry for SN1, but it reverses it for SN2 because of the nucleophilic attack.
Right. So SN2 is good, reverses. But what about SN1? So when you say no change, what do you mean? Now
Like,
think about it again.
oh, I was thinking it maintained the same stereochemistry as before, but it's not right.
No, it's not exactly right. So the first step is the departure of the leaving group, right? In SN1. And then the nucleophile, if you said there's no change, then the nucleophile would come in from the same side as where the leaving group just departed. But can it come from the opposite side?
Oh, it makes a, does it make a mix? Like it could be either way?
Yes, yes.
Okay, got it.
All right, so it leads to stereochemistry scrambling, right? So if it's an enantiomerically pure material, it will lead to racemization. It will lead to racemization. And so one possibility is when you have this carbocation shown here, right? When water comes in, it can attack from the top and can attack from the bottom as well. But only one product is formed where the water is on the top, right? Which is slightly inconsistent, but it is consistent with SN2, where basically this nucleophile came in from the bottom and totally blocked the bottom side. And so the water as a nucleophile can now only come in from the top, right? Only can come in from the top, kick out the leaving group, and generate this material. So, all right. Now there's one last topic that we have not covered today, but it's quite descriptive. So I just give you some reading. It's there's nothing really difficult about that. So how enzymes can be regulated. You can already see the main idea, right? So for example, with enzymes which are regulatory, you have catalytic site, C, and you have regulatory site, which is R. And let's say your substrate shown here, right? It doesn't fit into the catalytic site. It's triangle, and this is rectangle. So, but when the positive modulator binds to the R, now this changes its conformation. And when this changes its conformation, the catalytic site changes its conformation. So the two sides talk in each other. And now the substrate can come in and occupy the catalytic site, right? So that's the main idea behind the regulatory enzymes. And so just read about some examples, pages. Read pages 225. $235. Guys, are there any questions about anything we've talked about? Don't forget your sapling homework is due on Wednesday. And I'd like to see more discussion on Canvas. And I'll talk to you on Wednesday. Have a good couple of days. Okay.
Thank you.
Bye.